Welcome to this video. In this video we're going to be learning about cellular respiration. Now, cellular respiration is one of the first topics that students really start to struggle with AP biology because all these terms are introduced. You start to get these complex compounds that you start to have memorizing, but in this video we're going to try our best to make it simple for you. So, what is cellular respiration? It's simply the process of converting biochemical energy into usable ATP. Now, what is biochemical energy coming from? It's coming from our foods that we eat. So in the morning you woke up, perhaps you had a milkshake. This milkshake obviously contains a lot of biochemical energy. It obviously contains a lot of glucose, a lot of sugar which is not good for you, but this will ultimately be generated into ATP that's usable in our body. And of course, associated with cellular respiration, there's obviously a formula that you actually have to memorize, and it's glucose, C6H12O6 plus oxygen, which will give you water, it will give you some ATP, and also carbon dioxide. Now, this equation is not balanced. If you forgot how to balance the equation, go ahead and go back to our previous videos. Earlier on when we talked about the balancing equations in biochemistry, this will be C6H12O6 plus 6O2 plus 6H2O plus 6CO2 plus ATP. And this ATP stands for energy. So this is our main goal, obviously, and carbon dioxide is a byproduct. Right now I'm exhaling. As you exhale, you're releasing carbon dioxide. Also, if you don't know this yet, you're also releasing water vapor. So every time you breathe out, you're releasing some water vapor. We'll talk about that later. It's pretty interesting. It's a little bit disgusting, but we can learn about this later when we talk about it in our end video. So we start off with glucose. And we need oxygen. Where do we get oxygen from? Well, you have to inhale, and when you inhale, obviously you take in oxygen from the atmosphere, and we're going to utilize this oxygen somewhere down in this cellular respiration process. Now, to understand cellular respiration, it's broken down into three different categories, which you must know. The first one is glycolysis. The second one is the Krebs cycle. And the Krebs cycle is also noted as the citric acid cycle. And finally, the last one is called the electron transfer chain. Otherwise noted as the ETC. So, how do you go on about learning and memorizing this topic? Very simple. First, understand that cellular respiration is the breakdown of biochemical energy into usable ATP. We need three steps: glycolysis, Krebs cycle, and the electron transfer chain. Now, we're going to go into glycolysis. So now, let's start off with the first topic that we have to understand, which is glycolysis. Now, even before I talk about glycolysis, let's go ahead and kind of draw a picture to sort of get an idea of what we're doing here. So, here's a guy. And he's happy because he has just drank some milkshake. Obviously, this milkshake contains a lot of glucose. So there's a lot of glucose that's going down his esophagus and into his body. And this glucose is essentially going to be converted into a lot of different things by a series of glycolysis, the Krebs cycle, and the electron transfer chain. So, let's take glucose. This is our starting compound. Glucose. For those of you guys who don't know what glucose is, the molecular formula for glucose is C6H12O6. Recall, in biochemistry, we covered glucose. Glucose is a simple sugar. It gives you energy very, very fast. C6H12O6. This has 6 carbon compounds. Let's draw it out. It helps us understand this entire process. So we have a 6 carbon compound, and something very special happens to this glucose molecule over here in glycolysis. Now, the definition of glycolysis is the splitting of sugar. And essentially what you want to do is you want to split the sugar in half. You want to get the 6-carbon compound, split it into half. Obviously there are 12 hydrogens and 6 oxygens that I did not draw, but that's because I'm simply too lazy. Obviously we're just going to, for actually all these AP biology videos, if I use any organic compound, I'll just draw just the carbon compounds. So our goal here is to split this 6-carbon compound in half. Remember, glycolysis is the splitting of sugar in half. Well, how do we do this? Well, we actually have to invest 2 molecules of ATP. We actually have to invest 2 ATP molecules into this reaction. And what happens is you add a phosphate over here, and you add a phosphate over here, which causes the 6-carbon compound to split in half because it's highly unstable. Let's take a step back and talk about investing 2 ATPs. I told you cellular respiration is a process of converting biochemical energy into usable energy. It's a bit ironic that already we have to invest some energy before we can even make some energy. Keep this in mind. Before we can even generate any substantial amount of ATP, you must invest 2 ATPs into glycolysis. It's like in business. If you want to run a business, you have to first invest some money into it, and then finally, maybe 2 months down the road, or even 2 years down the road, you're going to start to see the money come in, and you're going to make profit. Over here, you're going to lose some ATP, and then finally make some profit. So by investing this 2 ATPs, you make this reaction highly unstable, which allows it to continue on. So now we have a 3-carbon compound bonded with phosphate. It's highly unstable, and it's eventually going to generate 2 pyruvate molecules. Now, I'm going to introduce a different term from pyruvate. It's also called pyruvic acid. Are they the same thing? Not exactly, but the AP Bio will use these terms interchangeably. So they can give you pyruvate, they can give you pyruvic acid. Just understand that they essentially mean the same thing. So glucose, your starting compound, will eventually give you 2 pyruvates. How does this happen? Well, you invest 2 ATPs. There's a lot of other things that go on here, but these videos are designed for you to only introduce things that you only need to know for the AP Bio exam. So let's go back over here. What do you actually have to know? Glycolysis. This is very important. In order for glycolysis to occur, it does not require oxygen. So, no oxygen required. Very important, cellular respiration. If you go back to the actual equation, we have oxygen. Remember, glucose and oxygen will give you ATP, carbon dioxide, and water molecules. Well, we don't actually need oxygen right away for this process for glycolysis. This can occur in oxygen or without even oxygen. It does not really matter. You must know this. Now, what else do we have to understand? As glucose is being converted into pyruvate, it's just not because of the ATP. There's also a coenzyme which helps facilitate this entire process, which is called NAD+. So there's NAD+ over here, there's NAD+ over here, and what happens is this gets reduced to a molecule called NADH. Now, once again, if you don't understand oxidation and reduction, please refer back to the biochemistry video. You should already know this. NAD+ will get reduced to something called NADH. Now, what's NADH? These are high energy electron molecules. These are high energy electron molecules. These are just like ATP, except does your body use ATP or NADH? Your body uses ATP. But what happens is this NADH will eventually be converted into some amount of ATP, and we'll talk about that later. But it's very important to understand right now that in glycolysis, with a single glucose molecule, we've generated 2 NADH molecules. Why do we care? Well, these are high energy electron molecules that are similar to ATP, which will eventually be converted into ATP, so we actually have to understand that now. The last thing that happens here is that you also get 2 ATP production from here, and also another 2 ATP production from here. So now already we're kind of happy. Look, we've generated 4 ATP molecules. However, the AP Bio test will always ask you for the net gain of ATP. What's the net gain of ATP you got in glycolysis? It's not 4, it's 2. Why? What does net gain mean? You've invested 2 ATPs, you've lost 2 ATPs, you've gained back 4 ATPs. So basically it's 4 minus 2, which is 2 ATPs. Once again, very, very simple math. So, what do we have here? A net gain of 2 ATP. And essentially, once again, I don't know why people find it complex or this topic to be, somewhat difficult to memorize, but this is what you really have to know. Glycolysis, definition of glycolysis, the splitting of sugar. What happens? You take a glucose molecule and you convert it to 2-pyruvate. How do you do that? You invest 2 ATP molecules. You get 2 NADH molecules and you get a net gain of 2 ATP. Understand also that this process occurs in the cytoplasm. So the location of glycolysis is in the cytoplasm. Let's do a quick minute recap. Okay, it's very simple to memorize. What do I expect you to memorize for cellular respiration for the part of glycolysis? Well, once again, glycolysis, splitting of sugar. You start off with glucose and you eventually make 2 pyruvates or pyruvic acid. There's a glucose, you have to invest in ATPs in order for this reaction to be unstable, so you can move it forward. You get 2 NADH molecules, you get a net gain of 2 ATP, and it occurs in the cytoplasm. It occurs in the cytoplasm. Before we end this section over here, let's discuss what the cytoplasm is. Here's your cell. Here's your nucleus. Here's your DNA. Here is your mitochondria. Please understand that almost the entire process of cellular respiration occurs in the mitochondria. So here's your mitochondria, and your mitochondria would be drawn like this. This is a powerhouse. It provides energy, but this is not your cytoplasm. Your cytoplasm is the interstitial fluid. It's all the space over here. So if there's a diagram on your AP Bio test and they're saying, where's the cytoplasm or where's the location of glycolysis, here is where glycolysis occurs, okay? So this is all you have to know. This is basically this is probably the most important things you have to know. No oxygen is required for glycolysis. We have a net gain of 2 ATP. The location is in the cytoplasm, and now we're left with our product of glycolysis, basically, which is 2 pyruvate molecules. Once again, it could be written as pyruvic acid. So go ahead and start memorizing this now. Okay, so now we're going to move on to the Krebs cycle, otherwise known as the citric acid cycle. Now, recall back for glycolysis where we got a product of 2 pyruvates. So we're starting off with 2 pyruvate molecules. So we have 2 pyruvate molecules. And now we have to understand how this 2 pyruvate molecules is utilized in the Krebs cycle. Now the first and most important thing here is the location of the Krebs cycle. The location of the Krebs cycle. Krebs cycle occurs in the matrix of the mitochondria. So, once again, these are the questions that the AP Bio loves to test you on. The location of each of these 3 steps. Does it require oxygen? Does it not require oxygen? What are your products? What do you get? What's your starting compound? What's your ending compound? So location is in the matrix of the mitochondria. Hopefully you guys know where that is. If not, let's draw a picture right here. So here I have my mitochondria, and here is my cristae. And inside, the space inside the cristae is called the matrix. So this is the matrix, this is the cristae, and this is the outer membrane. So, location of the Krebs cycle occurs in the matrix, over here. So, the most important thing you have to know also after the location is whether or not this process requires oxygen. And indeed, it does require oxygen. So this requires oxygen. So, after glycolysis, once again, it does not require oxygen. If we do have oxygen present, it will undergo the Krebs cycle, and it requires oxygen. What happens if you don't have oxygen? Well, we'll talk about that later, after we talk about the electron transfer chain. But Over here, let's assume we have oxygen. We can go ahead and carry this process out. Now, in order for this 2-pyruvate to actually enter into the matrix and start doing whatever it's going to do, what you need to do is first convert this pyruvate into something called acetyl-CoA. The reason for that is because pyruvate is a big, ugly compound. It doesn't fit in, and it won't go into the matrix of the mitochondria unless you oxidize this pyruvate into something called acetyl-CoA. CoA is the enzyme that transports this acetyl into the matrix of the mitochondria to carry out Krebs cycle. So this process over here where we convert the 2 pyruvate molecules into acetyl-CoA is called pyruvic oxidation. The process is called pyruvic oxidation. The most important thing you also have to understand from this process is that you generate 2 NADH molecules. 1 NADH molecule per pyruvate. If you're oxidizing this pyruvate, you're going to go ahead and produce some NADH. So you actually get 2 NADH molecules from this pyruvic oxidation. But that's important to know, sure. But we really care about this acetyl-CoA over here. This acetyl-CoA is now in the matrix, it's floating around, it's happy, it's just, waiting to undergo the Krebs cycle. And what happens is that acetyl is actually a 2-carbon compound, and it finds another compound called oxaloacetate. So oxaloacetate is also floating around in the matrix of the mitochondria, it's just waiting to find acetyl-CoA, and this over here is a 4-carbon compound molecule, and oxaloacetate and acetyl-CoA will come together to form a 6-carbon compound called the citric acid, which is citric acid. Once again, the Krebs cycle is also referred to the citric acid cycle. We're essentially trying to make citric acid, and we have made citric acid. We combine oxaloacetate and acetyl-CoA to form citric acid. What you might not know yet is that this is a cycle, so obviously there are other things over here. Luckily for you, you don't actually have to memorize any of the other intermediate compounds. You just have to understand, hey, acetyl-CoA, where do we get this? Well, we got this from pyruvic oxidation. After glycolysis, we have this acetyl-CoA. It binds with oxaloacetate to form citric acid. And then just some intermediate compounds that come, and once again, the same process occurs. This is called the Krebs cycle. Don't bother to memorize all the other compounds in between because the test will never ask you questions related to that. So, what do we have to know though? There are some things that you do have to realize about the cycle, other than what I've told you already. You have to understand the products. What's the point of this? We made some citric acid, great. But I don't really care about that yet. I want to focus on the products of the Krebs cycle. So, for each pyruvate which undergoes the Krebs cycle, you get some certain products. You get 3 NADH molecules, you get 1 ATP molecule, you get 2 CO2 molecules, and you get 1 FADH2 molecule. Now I've introduced a new term to you, FADH2. What is this? Well, it's simply similar to NADH. This is also a high energy electron molecule which will eventually convert itself into a certain amount of ATP. Well, actually, I'll go ahead and tell you how much ATP it makes right now. 1 NADH makes 3 ATP, and 1 FAD will make 2 ATP. So obviously there's importance in that, understanding why we actually have to focus on NADH and FADH2, but we'll talk about this more explicitly in the electron transport chain. So for each turn of the Krebs cycle, we're getting 3 NADH, 1 ATP, 2 CO2, and 1 FADH2. However, do we just have 1 pyruvate? No, we have 2 pyruvates. 1 glucose molecule gave us 2 pyruvates, so guess what? We're going to actually do the citric acid cycle, or the Krebs cycle, twice. So is the AP Biotest going to test you on this? No. It wants double these numbers. It's going to say what are the products of Krebs cycle resulting from 1 glucose molecule. And so I'm going to multiply everything by times 2, times 2, times 2. And these are the numbers you have to memorize. We actually get 6 NADH, we get 2 ATP, we get 4 CO2, and we get 2 FADH2. Once again, 6 NADH, 2 ATP, 4 CO2, 2 FADH2. Yes, you do have to memorize it. So, this is what happens in the Krebs cycle. Let's just summarize now what just happened. So, we start off with the Krebs cycle. We can't really use pyruvate in the matrix of the mitochondria. The pyruvate has to undergo pyruvic oxidation to generate acetyl-CoA. This is called the transitional stage, and we got 2 NADH resulting from this. Now, once acetyl-CoA is in the matrix of the mitochondria, it can go ahead and find oxaloacetate and bind to it. Oxaloacetate is a 4-carbon compound, acetyl-CoA. Acetyl is a 2-carbon compound, and together they form citric acid, a 6-carbon compound molecule. It goes ahead and you do all these intermediate things, and you keep doing this, and what you get for each turn is these products over here, but what you have to understand is 6 NADH, 2 ATP, 4 CO2, and 2 FADH2. Carbon dioxide is a byproduct. It's not a product. You breathe it out. Where do you generate carbon dioxide from? Krebs cycle. Now you know where carbon dioxide is really generated from. Location is the matrix of the mitochondria, and this process requires oxygen. So there's not much on the board, but whatever's on the board, you have to memorize for the AP Bio test. At the end of the third video for the electron transport chain, we'll have a series of questions.